Let's hit the ground running. Welcome, focused learners, to this essential explainer on Cambridge Biology 9700. Today, we're diving into something absolutely critical, the role of NAD and FAD in dehydration. You know the drill. This pops up in almost every single exam series, so nailing it down is quite literally your ticket to securing those top grades.
But here's the brutal reality we have to talk about right now. Why do students lose marks on multi-step biological pathways, even when they know the biology inside and out? Seriously, think about that for a second. You can spend hours cramming the exact sequence of dehydrogenation, you can know precisely how those hydrogen atoms transfer over to NAD and FAD, and yet you can still walk out of that exam room having dropped entire mark bands. It is the absolute most painful mistake to make, and it usually happens before you even realize what went wrong.
Listen to this. This is a direct, urgent warning straight from the principal examiner reports. Students forget to include units and calculations and fail to check terminology, losing easy marks. Guys, this is exactly the trap taking down top-tier students, right? Like, you know NAD and FAD carry hydrogen to the electron transport chain. But the absolute second you drop a crucial unit measuring a reaction rate with DCPIP, or slip up on a specific term, the examiner literally has to withhold that mark. It's a total tragedy of exam technique, not a lack of biological knowledge.
So, there are exactly 3 specific mistakes that just destroy grade boundaries here. 1. Forgetting your units. 2. Failing to check your terminology against the actual data provided. And 3. Giving generic answers. If the exam hands you a highly specific yeast respirometer setup testing dehydrogenase activity, your answer absolutely must anchor to that exact setup. You can't just recite textbook definitions of NAD and expect to get the marks.
Section 1. Core Principles of NAD and FAD.
Okay, let's dive right into this theoretical architecture. The whole study of NAD and FAD rests on precise definitions and, more importantly, causal relationships. It's all about cause and effect. It's like a giant puzzle. When a substrate is oxidized and loses hydrogen, how does that directly cause the reduction of NAD to NADH? You have to understand the whole system working together, not just memorize a bunch of isolated molecules.
So, the examiners are actively hunting for your proficiency across 3 specific cognitive domains. First up, do you actually have the knowledge and understanding of the core principles of hydrogen carriers? Second, can you handle and evaluate information? Like, can you extract and calculate data from complex graphs? And third, experimental skills. Can you identify anomalies in a practical experiment? If you fall short in just one of these, well, your score takes a massive hit.
Now, here is the absolute secret to bumping up a mark band: cross-topic connections. Dehydration is not some isolated little box you study once. It links directly, and I mean directly, to respiration, photosynthesis, and even the intricate structure of mitochondria. When you integrate those areas in your answers, you prove to the examiner that you really get the big picture. You might be wondering how to practically do this. Well, in just a minute, I'm going to show you the single examiner clue that completely changes how you answer dehydrogenation questions. But hold that thought, because first you need the framework that makes that clue super obvious. Without this framework, that vital clue is just going to slip right past you, and you'll be leaving crucial marks on the table.
Section 2. The Technical Method Framework. Meet your new best friend, the Deconstruct, Solve, Evaluate sequence. This is your ultimate grade-saving tool for navigating complex past papers. Step 1. Deconstruct. Look at the prompt, identify the given info, and the logical steps. Next, solve. You want to execute systematically with perfectly clear working. And finally, evaluate. Take a step back and ask yourself, is this reasonable? Does my biological reasoning actually justify this data?
And this brilliantly illustrates why knowing your stats is a total game changer. Think of this table as your statistical toolkit. Use a t-test when you're comparing mean rates of decolorization. Got categorical data? Go for chi-squared. Looking at relationships between variables like temperature versus dehydrogenase activity, you need correlation tests. Just memorize this setup right here, and you will save a massive amount of time in the exam.
Let's do a quick spot the error analysis to really drill this in. A typical, flawed answer just drops a final calculation on the page. No units, completely isolated facts, it's a mess. But a perfect top-band Cambridge response, it shows crystal clear working, verifies those units, and uses that cross-topic integration we talked about to explain the actual biology behind the numbers.
And if you're dealing with practical investigation design for dehydrogenase activity, this checklist is just non-negotiable. You have to select the appropriate apparatus, like a colorimeter, follow the sequential instructions flawlessly, make accurate observations, use well-labeled tables, and actively hunt for anomalies. Actually, scratch that. You almost need to anticipate anomalies before they happen. Without this strict sequence, any evidence-based conclusions you try to draw will completely fall apart.
Section 3. Evaluation and the examiner's secret.
Okay, now what's really interesting about this is an explicit guarantee directly from the examiners. Even if your final answer is wrong, method marks can save you. This right here is your ultimate insurance policy. If you write down your systematic working, a silly little calculator error on your very last step won't destroy your entire grade.
So, to secure literally every single method mark available, you've got to commit this standardized process to memory. 1. Identify the key elements. 2. Apply the relevant principles of NAD and FAD. 3. Execute your analysis with totally clear, legible working. 4. Verify your results. And 5. Present it all in the appropriate format. Remember, higher-order objectives demand that your final conclusions are absolutely justified by this exact sequence.
So here's the thing. You now have the framework, you understand the domains, and you've got the method mark strategy. But none of it matters if you misread the prompt.
Subscribe to our YouTube channel Ace the Exams at Ace the Exams IGCSE to excel in your Cambridge examinations. Because seriously, ask yourself, what happens if you perfectly execute this entire framework but misread the one single word that decides your entire mark band? What if the question asks you to evaluate and you merely describe? It completely tanks your answer. We'll demystify those tricky command words in our next explainer. Keep focused and we'll see you there.